Hello and welcome to section 17.3 on cellular defenses. In this section, we're going to talk more about the major cells within our blood, also known as our formed elements, and that would include our red blood cells, also known as erythrocytes, our platelets, which are also known as thrombocytes, and our white blood cells, also known as leukocytes. Now, our red blood cells are really used for carrying oxygen to our tissues, and our platelets are used for blood clotting and tissue repair. So our main focus in this section will be on our white blood cells, which are going to participate in a lot of our non-specific mechanisms within both our innate and adaptive immunity. But of course, in this chapter, we're focusing on innate immunity. And in this section, our learning objectives are to identify and describe the components of blood, which we already kind of talked about, and then to explain the process by which the formed elements of blood are formed through hematopoiesis, and lastly, describe the characteristics of formed elements found in peripheral blood, as well as their respective functions within our innate immune system. So we'll start off with hematopoiesis, and this is basically how we're going to produce our blood cells. And essentially all of our formed elements are going to be derived from this multipotent hematopoietic stem cell, also known as a hemocytoblast, that resides within our bone marrow. And a majority of these are going to become a red blood cell. So you can see that pathway over here through the myeloid stem cell. And then a bunch of other ones are going to become our leukocytes. Let me just touch, before we go into the leukocytes, let me touch upon our platelets. Notice how that is also going to be formed through the myeloid stem cell. And we first create this megakaryocyte, which you can see is one of the largest cells on this slide. And so we are going to break off little cellular fragments that then become platelets. So platelets are not truly cells as you think of them because they don't have nuclei. Anyway, let's move on into our white blood cells. So our leukocytes, you can see that they all kind of reside within this area here of our chart, and we subdivide them into granulocytes, meaning that they have granules that are visible within the cytoplasm, and agranulocytes, which means that they lack granules. We don't see them. So in this table, in this figure over here, you could see the breakdown of each of these cells and the number that's supposed to be present within our body, their appearance, their functions, and some other additional comments. So I won't go through this entire table, but instead we'll kind of walk through one by one of our white blood cells, first starting with our granulocytes, which remembers when we're going to see those visible granules within the cytoplasm. And this is going to include our neutrophils, also known as polymorphonuclear neutrophils or PMNs, our eosinophils, and our basophils. So starting off with our neutrophils, these are going to have nuclei that have about 3 to 5 lobes, and you can see they're connected with a little strand of thin material, and we'll see small, numerous lilac colored granules. So that's why they're kind of called neutral. These granules don't stand out like a bold color. Our eosinophils, on the other hand, are going to have these red large granules, and these are stained with eosin, giving that red color to it, hence its name. You'll see that they have fewer lobes in their nucleus. We typically see 2 to 3. And lastly, we have our basophils, which have 2-lobed nuclei, and they're going to have large granules that stain with the dark blue or purple, like a methylene blue. Let's focus more on those neutrophils. They're going to be involved with elimination and destruction of our extracellular bacteria. We'll see that their granules are going to have a variety of defenses that we've talked about before, hydrolytic enzymes that will help to destroy bacteria through the process of phagocytosis. So this is one of the key things to know, is that your neutrophils are phagocytes. They are cell eaters. They can also go through the process of degranulation, meaning they can release toxic molecules in infected areas. And we have extracellular traps that they create. That means that they have these meshes of chromatin that include proteins and can trap pathogens. And then we've got the ability to form pus, purulent, suppurative discharge, or drainage. And that really happens from them going through that process of phagocytosis. We start getting an accumulation of these neutrophils or leukocytes, which we call cellular debris, and the bacteria themselves. Now let's talk more about the eosinophils. These work to protect us against protozoa and helminths. Their role also is going to be within allergic reactions, and that's because their granules are going to contain histamine. We also have degradative enzymes and a compound known as the major basic protein, MBP, and that causes a disruption of the cell membrane and cell permeability of the microbes. Now with our basophils, they're going to work closely with our complement system. So they help activate complement fragments C3A and C5A, which act as anaphylatoxins by inducing degranulation of the basophils and inflammatory responses. So as you can imagine, these are going to be important in allergic reactions as well as inflammation. And we've talked about how their granules contain histamine, and that can help to open up gaps between cells in the blood vessels, so increases permeability. And it can also be activated by antibodies, so it kind of plays together with our adaptive immune system that really involves the production of antibodies. Now let's also talk about mast cells. They're going to be derived from the same common myeloid progenitor cells as the granulocytes, and their granules are going to contain histamine. So they'll have a similar role in the allergic responses and inflammatory reactions as the basophils. So I kind of call them best friends, and they circulate within the blood, but we mostly find them within our tissues. We'll also find them near our blood vessels and nerves, or in areas of our body that is close to an external environment, such as our skin and mucous membranes. Let's revisit our clinical focus on Angela. So they get the test results back, and for the allergic test, were negative for all the allergens. The sputum samples that we got, we found no pathogens or excessive microbiota. And the blood test, though, showed elevated levels of inflammatory cytokines in the blood and mildly elevated white blood cells, but normal antibody levels. And we also found that there's a lower than normal level of complement protein C4. So now with this information, we have to ask ourselves, what does this new information reveal about the cause of Angela's constricted airways, and what are some possible conditions that could lead to low levels of complement proteins? All right, moving into our agranulocytes. Remember here, these cells are going to lack visible granules within the cytoplasm. So there are two main agranulocytes. We've got our lymphocytes, which is going to include our natural killer cells, also known as NK cells, which play an important role in our nonspecific innate immune defenses, and our B and T cells. We sometimes call these B lymphocytes or T lymphocytes, and they're central players within the adaptive immune defenses. So we'll talk more in detail about them in the next chapter. And lastly, we have our monocyte here, which differentiates into our macrophages and dendritic cells when we leave the bloodstream and enter into the tissues. And collectively, we're going to refer to these cells here as our mononuclear phagocyte system, or MPS. So we'll start off with our natural killer cells, which are mononuclear lymphocytes, and they're targeting our abnormal cells. Something is off for some reason. It could be a cancer cell, or maybe it's infected with a virus. So it works to actively diminish or eliminate the expression of MHC markers, which then represents the cell as being abnormal. So MHC markers, you can think of them. Well, first let me say what MHC stands for. It stands for Major Histocompatibility Complex. And we'll see these markers within our healthy cells, or rather on the surface of our healthy cells, so that it can identify our body as being our own self cells. So it's kind of like a driver's license that says what state you belong to, right? So that we know that all of your cells belong to your own body. Now, it can also activate molecular molecules that can be displayed, showing that it is an altered self or non-self molecules. So if we don't recognize ourselves, we know something is wrong and we have to destroy it. So it's able to recognize those normal MHC markers on the surface of cells and serves as an inhibitory signal, preventing those NK cells from becoming activated. So when we see over here in this figure, this normal cell, it's saying, hey, I've got my ID showing that I'm from whatever it is, Natalie's body, it's going to recognize that. And notice how that activating receptor has nothing to attach to because we don't have that altered self or non-self molecule displayed the way we do down here when the cell is infected with a virus, right? We have that abnormal activating molecule displayed, and we don't see any of those MHC markers on here, and therefore we're going to destroy that cell. Now, how do we go about destroying it? We are going to express cytotoxic membrane proteins and cytokines that can induce apoptosis. So we use something called perforin-mediated cytotoxicity, which leads to apoptosis as well. And we are going to rely on a protein called perforin, and perforin creates a bunch of pores on the cell membrane that then will allow this enzyme called granzymes move into the cell, and that's a protease that's going to come into the cytoplasm of the cell and essentially cause the cell to lyse and die. Now let's talk about the monocytes a bit more. They're going to be the largest of our white blood cells, and it has a nucleus that lacks lobes. So you can see an example of some of these in here. And just like our neutrophils, they are going to be effective phagocytes, although they kind of come into play at a different time point during the inflammation process. So we know our monocytes circulate within the blood, but once they leave that bloodstream and enter tissue, they differentiate into tissue-specific phagocytes known as macrophages and dendritic cells. And our macrophages, they can be given different names if they are in a specific tissue. They're kind of like resident macrophages, and you could see some of those listed here on the right. We see in the brain and central nervous system, we have our microglia that are macrophages. Within the liver, they are Kupffer cells. In the lungs, these are my favorite names, dust cells or alveolar macrophages. And then we've got our peritoneal cavity that has our peritoneal macrophages. Now, as far as our dendritic cells, those are going to remain mostly within our skin and our mucous membranes.